All right. Good evening, everyone. Dr. Prasanna. I thank Dr. Ketana Barcode, CEO of Barcode Biotechnology, for this beautiful invitation. I've been working on bacterial identification for more than 15 years now, and I've been associated with Barcode Biotechnology for a long time now. They are during my master's dissertation, PhD till now, they've been providing me with various services, especially sequencing services. So, today's webinar is all about identifying bacterial species using DNA sequences. And I will not be talking much about the theoretical part involved in this because we have a lot of resources to know everything now these days. We have AI. So I will be talking about business parts and the technical part of practicality. What are the things you need to be focusing on when you are actually identifying the microorganisms, okay, especially bacteria. So it's my obligation to introduce Barcode Biotechnology. They are most affordable sequencing service in India, and they offer various other services. Basically, they do Sanger's and Illumina sequencing. They are founded in 2013, and now they are more than 12 years of their existence, and they are the most affordable sequencing service providers in the country, I would say, in India. Apart from that, it's not just business, they have an ambition of 1,000 barcodes initiative where they try to barcode a not yet barcoded species. Okay, what is this barcode? It's a DNA fragment used to identify the organism as belonging to particular species. Okay, why do we need to sequence and have a library and all other steps? Because only when you have an enhanced DNA barcodes covering more species, you can use the barcoding as a technology. Call it as barcode technology. We will see all those things. I will quickly show you the company's infrastructure before getting into the sequencing part or identification part, especially. So we have a ABA sequencer, we have a megabase sequencer, PCR workstations, and there is a Mermaid system which produces primers. It's a breadth supplier of barcode biotechnology. So related to bacteria, Barcode Biotechnology gives bacterial identification service. They give a mixed culture identification service. You can simply find out which colony you want to sequence. They can do the pure culturing and go for sequencing. They have something called actinobacterial identification service and fungal identification service for all bacteria, both archaea and bacteria, all prokaryotes. Actually, 6NS is a gold standard for sequencing and identification. An internal transcribed spacer is a region. For fungi, it is used as a gold standard for identifying a fungi. Cyanobacteria, 16S. Mushrooms are macrofungi, so ITS. They are also doing phage identification service. Whenever you see a plaque in the Petri plate, you can simply make an buttons and you can send the phages, send the agar buttons. The Barcode biotechnology are equipped with isolating the and purifying the phages and going for sequencing. They also provide something called yeast identification service. Yeast again being a fungi, ITS regions are actually targeted. They have a full bacterial characterization assay because just with 6NS rRNA, many times you may not be able to climb a particular fragment belonging to a particular bacterial species because there could be a horizontal uh, gene transfer. Okay, many times 6NS rRNA, multiple copies, single copy can be transferred, and there are possibilities. So whenever you propose a particular species, you need to give them a justification with their biochemical complements, biochemical characterization. For that, Barcode Biotechnology actually they do initial 12 to 15 assays to guess what genera it could be, and then based on Burgess manual, they will go for sequencing. After sequencing, they can also do reverse taxonomy, meaning, say for example, if that sequence is 99.8% similar to Gluconobacter nicotinae, again they can design an experiment to prove that it is a nicotinae species. Few biochemical will be proved and that can be done. So something it's unique in the country, this service. They're doing around 27 assays to prove a particular bacteria belonging to a species. Okay, they are also doing functional gene analysis. For example, if you have a bacteria which is resistant to multiple drugs, multiple antibiotics, if you want to see what kind of genes are being expressed, you can avail this gene sequencing service. They are also doing this cDNA extraction, cDNA synthesis from RNA extracted and multiplex PCR to see what are the antimicrobial genes that could be expressing. So let's quickly get into the topic. How to identify a bacteria. You should know in the biological science, we know now that DNA is a genetic material because of bacteria. Okay, starting from Griffith experiment, who used the streptococcus, passing on to Avery McLeod, Hessian's Chase, Fudge experiment. Every experiment has actually used microorganisms to know what is the nature of our genetic material. Okay, so having said that, bacteria, whenever we say that the father of microbiology is Adam Leeuwenhoek, so there are many people here. We are listening to this microbial bacterial identification service for first time. So I will take few minutes to talk about basics of what is this bacteria. Those belongs to a prokaryotes and they can be cultured as a colony on the surface of agar-agar. Agar-agar is something solidifying agent you add to the nutrient soup consisting of beef extract, yeast extract, then agar-agar. One of the main, no one will realize this as easy. The main quality of the media to harbor bacteria is its transparency. Okay, if the media is not transparent, you will not be seeing a colony. So the individual mucoid circle, what you are seeing is millions of bacterial cells. Once it was a single cell before 24 hours, then that becomes a millions of cells based on the physiology and speciation. They may take various forms. Each and every species, most of the species have their own forms of growing colonies. So those are called as colonies. You should know it's a big business happening in India. The bacterial identification service is estimated to be around $8.2 billion near to 2030. Okay, so it is not just a taxonomical formality. It's a foundational step. You will see that determines many things, including accuracy, safety, and efficiency of applications in various sectors. So before getting into species identification, this word is very important because the conventional taxonomy and the using microscope and assessing the molecular, morphological characters and biochemical characters, they usually you cannot differentiate the closely related species. When I say species, I just want to remind you guys about domain, kingdom, phylum, class, order, family, genus, species. This is a taxonomical hierarchy. So when you want to compare, say for example, the probiotic Lactobacillus delbrueckii belongs to Lactobacillus genera, which belongs to Lactobacilliaceae family, Lactobacillus order, Bacilli class, and Firmicutes. Likewise, we have proteobacteria, we have Lactomycetes, various phyla of bacteria. Like we have various phyla for Homo sapiens, under for Mammalia and many other groups. So I want you guys to remember this uh, three kingdom concept. See, we all belong here. All fungi, including humans, are in eukarya. Bacteria as a separate kingdom and archaea as a separate kingdom. Together, they called us prokaryotes. But they used to say the genetic difference between human and elephant is the genetic difference between bacteria and archaea. Even though both are microscopic and unicellular, you should not consider them as the same. So today's webinar is all about bacteria, how to identify a bacteria. So when it comes to microorganism, we don't stop just with the species. Many of you have listened to subspecies, zero was. So a species may have a group of similar looking strains, you can say. So what is this strain will come into? So group of similar looking species are subspecies that are unique in their own ways. Okay, example, in Salmonella under Enterica, the typhi and everything comes under Enterica. So Salmonella typhi, the bacteria that causes typhoid, actually typhi is not a species name. Salmonella enterica is a species name under which the subspecies is typhi. So what is this zero war then? Sometimes, or in many times, even the subspecies can be categorized based on antigen antibody reaction. So it is more precise to clinical pathogens. Okay, they cannot be applied. Zero was or terminology is not applied in environmental microbiology. So they are precise to clinical pathogens. And what is this strain? Strain is something you assign. The alpha numerical values you assign, you can give a strain. Each and every unique variation in the bacteria should be recognized as a strain. I hope during COVID you have seen many strains, even in one nucleotide substitution, they will be regarded as a particular strain. Okay. So what are the all right. So when you draw a phylogram for species, subspecies, and serovars, this is how it is going to look. Similar, serovars, they clump in one clade under one subspecies, say here typhi, which is under the Entrica. So all typhis will be cladding here. If there is a small differences in the genome, they can be placed in the another subspecies. So in basically in the phylogram, if the two species are placed in same branch, they share more similarity. If one species, say for example, Salmonella enterica is placed away from E. coli, it means they are genetically distant. Okay. So in among these Salmonella enterica and Bongori sequences, when they introduced E. coli, you see the E. coli branch has been cladded out and Sigilla also. So we will see more about it. So how does a bacterial taxonomy starts? Usually it starts with morphological characterization where you measure the individual colony, say size, shape, texture using staining technique, and then you will go for various biochemical assays to understand the physiology of bacteria. Or in other way of saying, actually you are understanding the genome of bacteria through biochemical, okay, of various biochemical assays. So as I said, sequencing is a gold standard in bacterial taxonomy. If at all you want to propose a species name, you have to go for DNA sequencing. In other way of saying, if I say even E. coli that is giving metallic sheen color in an eosine methylene blue agar, still you need to have a 16S rRNA data to support that it is an E. coli. So what is this 16S rRNA? So 16S rRNA is 1,500 base per region. It's a code for ribosomal RNA that plays a platform in protein synthesis. They form a ribosome complex and play a vital role. So whenever here is the length of the 16S RNA. Whenever we talk about 16S RNA, especially in next gen sequencing, you will be talking about V1 to V9 region. What is this V1 to V9 region? So among the 1,542, this is from E. coli's genome, it's around 1,500 base pair. So they have discovered 9 hyper variable regions which can be used. So why not we use the entire length? Why to have a hyper variable region? Simply because of the technology. Okay, Illumina platforms, they have various platforms like ISEC, MySEC, but what is happening is they just produces around 300 base pair and 450 base pair. But our 6NS is 1,500 base pair. So instead of, but they can give you more sequences. For example, one bacteria, you can even end up getting around 7 million sequences of their 6NS. That whatever is being amplified in PCR, every segment of amplicon can be given. For example, if I give for my sec, I may be receiving around 2 to 3 GB of data. And each and every raw file, when we are subjecting them to further analysis, will generate more space, more data in the system. So this is a technological limitation that made us to classify 16S rRNA into various hypervariable region, but that's okay. Nowadays, Novasec have revolutionized this thing. Even the whole genome of bacteria can be obtained easily. So why only 16S? Is there any better gene that can identify bacteria? So what are in other question, what make a gene to act as a taxonomical marker? Any gene that has a variation, conserved variation or much similarity within the species than, more differences between the species of same genera, then that gene can be used as a barcode. Okay, I will put it in very simple words. The intra-species variation should be less than interspecies variation. Intra means within species complex. Say for example, Salmonella enterica. The variations within that particular gene, okay, inside a particular species should be less when compared to Salmonella enterica and Salmonella bongori. Two different species of paratyphi, two different species of Salmonella. The variations, when I say variation, what I meant is if there is a 100 base pair DNA fragment, how many base pair it is differing, in how many bases it is differing. Okay, so say for example, in 100 base pair length, If I'm having 10 nucleotide differences, I would say it is a 10% variation. So in bacterial taxonomy, the general cutoff is 3%, meaning in 1500 base pair, 3% variation is allowed to call a particular sequence as the same species. So what is this species variation? Where are we comparing to? So how this entire sequencing works is you sequence a known species, put it in a database. When someone who do not know what is their bacteria, if they sequence it, they can compare their sequence with the database. They will know what species it is. That's how the molecular taxonomy or gene-based taxonomy works. Okay. So that's why we have a blast analysis in NCBI where you sequence your DNA and you put it in blast. When the blast compares the query sequence is what we give, and reference sequence is something that is there in the database. If the percentage of similarity is 99% similar to E. coli, then the sequence is E. coli. But if it is 98% or 96% similarity with E. coli, maybe it is an Escherichia, but not coli. Why? Oh, it's only 96, meaning 4% variation is there, but the cutoff is 3%. So anything equal to 97 or above 97 is actually same species. Anything less than 97, 96 or 95, maybe same genera, but not that particular species. Likewise, there is family cutoff 93, and we have a class cutoff around 89. So every level of hierarchy have a cutoff. Okay. So does it mean that yes, 16S rRNA qualify these all characters, but still we have another gene called RPOB. Okay, this RPOB actually has proven much more better resolution than 16S rRNA, but still we use 16S rRNA simply because we already have filled the database, invested heavily in producing this data, and created a mega databases like DNA database of Japan, EMBL, European Molecular Biology Laboratory database, and National Center for Biotechnological Information, US database. We have flooded with 16S rRNA. How much is accurate is another story, but we have flooded with 16S rRNA, and replacing this entire system of classification now takes a lot. So even though RPOB, RNA polymerase beta subunit gene of bacteria is known for better resolution and better molecular marker than 16S, we still cling on to the 16S rRNA. Okay. So that's why sometimes for precise identification, there is something called multi-locus gene sequencing where people are advised to sequence both 16S rRNA and RPOB. RPOB is around 2,500 base pair, and 16S is 1,500 base pair. Together, they can form a good team. But even then, if you are getting, the similarity is 96%, that's when you should know you have a new species. Whenever a particular reference sequence analysis exceed the proposed cutoff, that is 3% in case of bacterial species, that's when you know you have a new species. Then we have average nucleotide identity, DNA hybridization, entire whole genome sequencing should be done in order to propose them as a new species. So basically, bacterial identification work by 4 stages. Okay, the stages are DNA isolation, and the entire bacterial DNA is isolated. Then they are subject to PCR, where a particular gene is actually selected. 16S primers are used to select 16S, and RPOB primers are used to select RPOB gene. Then there will be a two-way sequencing, and then we will have a sequence analysis to know which species it is based on the sequencing. So how this entire system works, we generate huge amount of data, we deposit in libraries like NCBI, even India has a library, we will see that now. NCBI and many other databases, and you assign them to a particular species so that when someone who do not know what species he has, he can sequence the same sequences, same gene here, the 16S rRNA, and compare it with the database. Okay, so all this is fine, but these days Illumina, the next generation sequencing, has swiped away all these requirement of single sequences from a specimen. They are producing millions of copies. We will see them one by one. Okay. So this is what it does. You guys know that most of the bacterial species are unculturable. If I take a gram of sediments or soil, 99% of species cannot be cultured. Okay. See, only when I know a total number, I can propose a percentage. What I meant is, Only when I know a total number of something, if I know that there are 100 species, only one species form a colony, then I will say 99 species is unculturable. But how come people are always saying that 99 is unculturable? Simply because of metagenomics. You just extract the DNA in the PCR, you use your 6-ness amplicon and go for sequencing everything. 10 million reads you may end up producing. So the another immediate question from the young minds could be, if I have a single sequence, I will go for blast analysis. If I have a 10 million sequence, how can I know the taxonomy? So these databases give something called reference files. You may have seen it here. Okay, GenBank, Silva, RDP, ribosomal database project, they all have a reference file, meaning your sequencing file along with the reference file can be merged and the analysis, taxonomic analysis will be done. For example, the results are represented like this. Here it is a phylum level taxonomy. How many acidobacteria, bacteria, cyanobacteria is there? They are represented. Here it is a genus level taxonomy. What are the genus and how much it is there? Usually the size of the bar usually indicate how much it is there. This is a basic work. Even though I'm talking about metagenomics, this work is all about using various preservative agents to see which recover more diversity. Okay. So like I said, once then there is an average nucleotide identity. You have to compare the nucleotides to know how much percentage are similar or different. And there is another scenario. So does it mean that always 6-ness rRNA and other this 3% cutoff works for everyone? No. Okay. Every species, every genera, in my opinion, should be having their own cutoff. Okay. So if that is 3% cutoff can be simply used as a guideline for seeing whether we have a new species or not. Otherwise, see here, E. coli and Shigella are known for their close genetic similarity. Okay, and you introduce a Shigella dysentery, they actually form a same clade with the E. coli. This is not okay. This is the fate of 16S rRNA. So there are closely related species that require alternative genes for identification. Okay. Uh, what is this? Before I get into showing you the practical aspects of what we do in Barcode, biotechnology, how to classify organism. I will show you this how this artificial intelligence is actually revolutionizing bacterial taxonomy. The great plate anomaly is nothing but, see, if I plate a gram of soil and have a 20 unique morphotypes from the soil, okay, only 20, but when I take my DNA sequence from the soil directly and went for, I may have 200 species, but in the plate I have only 20. Okay, now I want to compare my DNA sequences of bacteria that formed a colony in a plate with the metagenomic data. If I compare my DNA sequence, none of the sequence will match with the sediment data, sediment DNA sequence. It's not logically right, right? This is called great plate count. Maybe there is a dominant organism who never form a colony. Maybe there are resident, minorities whose population is very low. They may form a dominant colony in the lab condition. So it's a sorry. This is a great plate anomaly we have. Okay. So there is another study I want to highlight. Barcode Biotechnology, as I told, it's the most affordable sequencing services, charging 1,850, I guess. This time they are charging 1,850. You can send a live sample and could get it. But actually, the cost can be go near to 4,000 or 5,000 rupees per sample. I know many of you, people who are constantly working with Barcode Biotechnology, students and research scholars will know this. It's the most affordable DNA sequencing service. And this may even become cheaper because of AI. Here, what they are trying to do is they are trying to study the images of various bacterial colonies. They are trying to take a picture of a bacterial colony. They are trying to study its images, and they try to build a big data so that simply by a time comes, we can have a trained model. Simply by clicking a picture of a bacteria, the species of the bacteria can be relieved. Okay, revealed. Sorry. So how this is possible is maybe each and every pixel in the images, they say something that we cannot visually observe. Okay, so each and every picture is converted into numerical values, and a picture under different intensity of light in different media. If you train the model well, you may have a workable, functionable model. So this is a proof. One paper they have screened more than 10,000 bacterial samples belonging to more than 100 and something bacteria. And they said the accuracy of species detection just by taking a picture is 95%. So simple, with a simple picture, the model can predict what species of bacteria it is. Yes, the time is coming. So how does this work is they usually streak it, have an isolated colony. Each and every colony, okay, they document it and they convert the value. For example, there are two colonies here. Definitely the pigmentation pattern is different. So the pixelated value, the image values can be converted, for both the colony margin and colony pigmentation, they are completely different. These model can be trained like this. You just sequence and know the bacteria and feed the images also. So maybe this is E. coli, this is Serratia. So whenever you show them under the AI models, they may instantly recognize E. coli and Serratia if the model is sufficiently trained. Okay. So good times are coming actually, and even we have used a similar model here. For long back, copyport bacteriobiomes are nothing but both culturable and unculturable diversity in copyport is actually a small crustacean in the marine system. So there was a talk that copyports all have same bacterial diversity in their gut region. Another talk was that every copypod will have species specific bacteriobiome diversity in their gut region. So when such a talks are happening, we thought we will apply this machine learning tool from Kime software to predict whether they really have a core operational taxonomic units or accessory. So what is interesting is it is true in both the cases. Every copypod have a core bacteriobiome that does not change between and within copypods, but every copypods also have accessory bacteriobiomes that change with different species of copypods. So again, here is an AI's performance. So what we do I want to take this mixed culture identification service as one example to demonstrate how the bacteria is identified in the labs. Okay, it's not a micro, it's not an animal or plant. Simply, you can see it. We are going to do a taxonomy of something you cannot see through your naked eyes, unaided eyes. Okay, so this is what makes this entire process so exciting. So every taxonomic classification, in my point of view, will have 5 steps. First will be macro and micro morphological characterization. What is this macro? Colony characteristics. How big is the colony? What is the pigmentation margin? What is micro? Cell level characterization. What is the volume of the cell? Gram positive or negative? What is the shape of the cell? Cocci or rods? So cell level is a micro characterization. Then comes at chemotaxonomy. We will be exploring the genome of a bacteria through various biochemical reaction to check its presence and absence. Simply put, this is what the chemotaxonomy, a biochemical characterization means. The third step is molecular taxonomy. Okay, where you can use polysaccharide profiling or even protein profiling to see how many different types of bacteria you have, and then you can select them for sequencing. Then 6S rRNA gene sequencing will tell you what kind of species it is. Fourth step is reverse taxonomy. Actually, you should see how the proposed species through 6S rRNA gene sequencing can be complemented with the biochemical reactions. Unless until you will see many papers, say for example, they may have 99.9% similarity with the lactobasilus delbergi, so it is lactobasilus delbergi. But the paper, the manuscript will say it is lactobacillus species. Without the supporting evidence for delbergi in the biochemical reaction, you cannot propose the delbergi. Okay, so I would say this is something called reverse taxonomy. Even after the 6NS data says so, because there could be chances of lateral gene transfer and many things are there. So you have to support it with the reverse taxonomy. And then comes the sequence analysis, where you will, altogether propose a particular species. So morphological analysis, biochemical analysis, molecular analysis, reverse taxonomy, sequence analysis. All together can propose a particular bacterial species. So in Biotechnology, imagine you are working in the sequencing company, you receive a Petri plate. So I will take you from there. Okay. I will take this mixed culture identification service as an example. Even a plate as poor as this. Okay. Even though you don't have a isolated colony, perfect coherent streak, now the client needs to identify all the bacteria here. Okay. So what we will do, we will analyze the individual colony morphology, Okay, if I may, I will take this colony as a unique one, and the round colony without irregular margin. All unique morphotypes you identify and go for quarant streaking. Okay, so we'll have something called work plan. Once we get the plate, we are supposed to give the results in 15 days time to the clients. So we'll make the work plan. This is how the plan goes. Okay, this is a first FIR we do in the lab. When you receive the unique ones, the clients will mark something like this. This colony 11 and 18. Please sequence and let me know. And also give me the pure cultures of 11 and 18 after sequencing. So once after sequencing, once you know the what species it is, you are supposed to have the bacteria in order for further exploration. So they are giving the pure culture for free, the Barcode biotechnologists. All right. So pick the bacteria, go for quarant streaking. The objective of quarant streaking is to get isolated colony in the last quarant. Probably go to broth inoculation, then make a glycerol stock, revive the stock to see whether the quarant streaking is working or not. If yes, again, then go for it. Two more stocks. So we would usually do per sample 3 glycerol stocks. Everything documented very precisely. Then we will go for gram staining, whose objective is not only staining the cell, but also individual cells can be very clearly seen. I would advocate gram staining as a biochemical technique instead of just microscopical, because it will tell you the cell wall composition. Here we can simply say that peptidoglycan layer is more than lipopolysaccharide because the purple stain has been retained even after ethanol wash and saffron. It is not been counter stained. Since because they can tell us the cell wall composition, I can advocate it as a biochemical technique. So we have oxidase test, catalase. We are looking for particular enzymes that can convert hydrogen peroxide into water. Here they are detecting cytochrome C oxidase using the dye. We will do a motility check. Broth characteristics, are they growing is a pellicle at the sediment? Then finally antibiotic sensitivity test. So why to do antibiotic sensitivity test? Even we use these characters to identify unique ones among this population. What are the unique ones we can select for sequencing? Usually we subject the gram negative rods for in-week assay, and we also do spore staining to see whether they're spore relative or not. Both gram positive and negative will be subjected to TSIS test, and gram positive will be subjected to starch hydrolysis. Okay, likewise, many more assays. From when we complete this flow, we will have an idea what family of bacteria we may have. Okay. So after this, there is a numerical taxonomy where each and every value can be converted into ones and zero. For example, gram positive, negative, all positive, negative, negatives can be zero, positives can be one. Finally, if you have a 10 bacteria, prepare a numerical value for all the 10 bacteria, sum of all scores. Just calculate for all the bacteria divided by number of assays. Each and every bacterial strain will have one unique numerical number. Usually, the unique numerical values can be a unique bacteria. Okay. In the repeated values, you need to check the data again. So what I will do, I will just show you various pictures of how the documentation is done here. Okay. So isolated colony from the mixed culture, this is the first step. This is the way to do the quarant streaking. We usually probably say this is something called 6-3 cascade streaking. What is mean? What is it mean is just make a quarant here, 6 or 8 lines. The subsequent quarant will start from last 3 lines of the previous quarant. If you have this one policy fixed when you are doing a quarant streaking, definitely you will get an isolated colony in the last quarant. There is something called last quadrant metrics, which is very much essential in bacterial species differentiation. Okay, metrics usually mean measuring the size of 7 to 10 colonies and take the average of the last quarant metrics. Okay. I know many people do continuous streaking, discontinuous streaking. I would say 95% of time you will get an isolated colony if you go for this 6,3 cascade streaking. This is not the way to do the streaking. Inoculation loop may be so hot here, hot here. The background is not okay. You should always take the wrapper out. I know there are many debate about using the wrappers in the petri plates. Okay, is it okay to use the wrapper or not? I would say in academic setup, you have to use the wrapper. In the business setup, maybe it's not essential. Okay. Why only in academic? They will prevent contamination. The incubators are actually stacked up with the plates. When you open the incubator, they are supposed to slide off and come away. So if you have a wrapper around this, something like this, it acts as a friction. They will not, that prevents the damages. But many experiments have proven that there is no growth difference between wrapped plate and unwrapped plate. Okay. Those micro volume of air that is present inside is sufficient for their survival. This is also a quarantine sticking, not a contamination, I would say. These organisms grows like this. They swarms, they form a vinculal undulated marginal colonies. So that's how they swarm. That's how they grow. Okay. Here also, this is like I'd say this is a bacterial intelligence. You see the quarantine streak, only the outer streaking lines are actually swarming. The inner lines do not superimpose on each other. This is still a pure culture. Okay, and this is an exhibition of bacterial intelligence. All right. So it's okay. Even if your bacterial culture swarms, just increase the concentration of agar-agar. Usually 1.5% is used for solidifying. If you increase to 2% or 2.25%, you will see the water activity of the media will be reduced so that the bacterial colonies will not swarm. Okay. So once you are very much thorough with the inoculation loop, you can play like this. All the odds are actually represents a bacteria with the different pigmentation characters. Here is a Louis Pasteur, actually Streptomyces casei, that is being streaked in Actinobacter isolation agar. Okay, uh, that's a very good art. So the first thing you do after last quadrant matrix is to study the size, shape, and elevation of individual colony and classify them like this. Many colonies may also have multiple characters. For example, they may also have lobate and undulate margins. Okay. So the first thing is gram staining, where you have to visualize individual bacterial cells. Okay. The gram staining should ideally be like this. Individual cells should be clearly visible. Usually, if you prepare the smear precisely at the edges of the smear, usually you can see thin films of bacteria distributed everywhere. Not like this. This is also a gram staining. You will not see, you will not interpret anything out of. Likewise, spore staining, the right way to document. The green color structures are actually spores within and outside the cell, and the saffron stained cells are looking as pink color. Then the capsule staining. Okay, this is the way to do, and this is not the way to do. All right. Then slowly we can go into biochemical test. Every biochemical test should be supported with the uninoculated control. Following incubation, both the tubes should be added with the necessary reagent. Both should be documented like this. Okay. So uninoculated control, white background. Even though these pictures look very precise, why it is not okay to have such a picture? There is no control here. Uninoculated control is very much important. Then you can subject both gram-positive and negative bacteria to triple sugar ion. What are those three? Sugar, glucose, sucrose, and lactose in various concentration. This is very simple test I like most because it tell you more about the aerobic fermentation nature of your bacteria at the same time anaerobic fermentation nature. Okay, how this is done is you can see the unusual height of the bud. Okay, usually the bud is around 20%. It starts from 20% length, but here the in TSA it should start from 40%. So the cultures, bacterial cultures are stabbed. When you are pulling the needle out, then they are streaked on the surface of the slide. The surface bacteria growing here actually is undergoes aerobic fermentation. The bacteria here undergoes anaerobic fermentation. So one simple assay will tell you what kind of acid or alkaline is produced from the what kind of fermentation that is happening, and is they producing hydrogen sulfide gas or just a colorless gas, okay, that you can lift the bud here. So many information can be derived out of triple sugar ion. Okay, so catalase assay, looking for the enzyme catalase in the bacteria. You can simply pick the bacteria in the wooden prick. Usually metal loops are not allowed because sometimes even loop will have a degradation activity of the hydrogen peroxide. So when you dip a bacteria, if the bacteria has a catalase enzyme, you will see effervescence of oxygen is released. H2O2 plus on catalase action will give rise to 2H2O, water plus O2. These O2 you can see as a bubble effervescing out. Okay. Then we have a disk for screening for oxidase, cytochrome C oxidase, and a respiratory chain bacteria. So those discs, if you can inoculate your bacteria within 10 seconds, if it is turning blue, it is positive. Not turning blue, it is negative. Turning blue after 15 seconds, pseudo-positive. Okay. Then you will check for the motility. The bacteria has a flagella, which can be used to move in various directions. So through hanging drop method, you are actually checking for the motility. Like I said, you can also subject the bacteria to various antibiotic sensitivity assay. Here, chloramphenicol, ampicillin, streptomycin, everything has been used, nystatin. So these bacteria is resistant to 3 or 4 antibiotics. These values, you can simply measure the zone formed by the bacteria, and then you can compare the zone with the standardized table. Okay. This is not the way to give the seed culture. Okay. Because the seed is not uniform, the broth is contaminated, many other issues. So after that, you can measure it, compare it with the standard table, you will know whether the bacteria is resistant or not. So this is something unique we follow in Barcode Biotechnology. When we have 10 bacteria in same sample, if you want to know whether the 10 has to be sequenced or not, because it costs. Okay, bacterial DNA sequencing costs us. So okay, sorry for the glitch. So here, what we do is we will simply dissolve the bacterial biomass in the loading buffer, and we will just load it. Okay, this is STS polyacrylate gel electrophoresis. We use Bowen serum albumin as a standard. You will see based on the number of bands, we can compare the bacterial growth. Okay, I definitely say this is a different bacteria and this is a different bacteria because the banding pattern is completely different. The banding pattern usually tell us the whole cell protein profile. So any bands that are more than here are actually has more molecular weight. They have more than 68 kDa for sure. Anything below here is less than 68 kDa. That's why they have moved faster. Just based on the fingerprinting pattern, we will know how many bacterial types are there in our culture. Okay. And also here, we will isolate DNA. This is the first step in DNA gene sequencing. Then we will go for PCR, amplify only the 6' rRNA and go for sequencing. So we also follow something called taxonomical heat map, where each and every character, positive characters are indicated by, this is the strains that we analyzed, and the positive characters are indicated by green and negative characters are by blue, by red. Comparing green and red, we will know how many unique morphotypes are there. Only those unique morphotypes are subjected to sequencing. Among 20 different morphotypes, we may have only 15 or 16 bacteria. So those repeated species, you don't need to sequence, just go for the unique ones. These days, we will have API strips. Many people asked me, what is this API? They are ready-made strips for various biochemical assays we have seen now in wake citrate. Many assays can be done through these strips. That bacteria is inoculated here, incubate the strips overnight. You can see the results. So why do we do all these biochemical assay, morphological assay? What is it's required? What are you trying to prove here is, this is the Burgess manual, an authority of, uh, there are multiple volumes. It's an authority for bacterial classification, especially bacteria and archaea. So all the 30,000 bacteria and plus bacteria that is being discovered, their characteristics are clearly documented in Burgess manual. Okay, so we are trying to create data through biochemical and molecular analysis, and we are going to compare it with the Burgess manual to see uh, if we are matching with something. I would say based on this molecular data and biochemical data, not molecular, biochemical data, mostly family levels, where general level identification can be derived through Burgess manual. Then comes your molecular data for species level identification. So these are the practical aspects of bacterial identification happening in our company. So I want to show the basic services again. What we do other than microbial, okay? They have animal species identification service. You can send us a tissue of plant. I'm sorry, fishes, mosquitoes, insects, lizards, anything. They can identify what species is it through CY gene sequencing. You can also send us uh, any other animal samples or even insect samples under insect barcoding. Cytochrome C oxidase subunit 1 gene is being used for barcoding animals, even marine samples, community samples. You don't need to have individual samples. Even if you send us a community samples, we will tell you what are the community of organism present in the sample. For example, biofouling community that may have more than 20-30 species. If you scrap off with the community and preserve an ethanol sent to Barcode Biotechnology, I think they are charging 1,850 per sequence. They will per species actually. They will tell you how many species they have. And these days, plant identification. You can send the plant tissue samples. They can simply barcode the RBCL, MATK gene. These are the barcode genes for plants, and they are used for authenticating of herbal formulations, even herbal powder. If you want to know whether what they have advertised is the same plant they have used for preparing the product, it can be cross-verified by extracting DNA directly from the product and identifying the plants. Okay. And in forest survey also, these days, instead of just tracking animal footprints, even if you have a fecal samples, you can decode what animal the whose fecal belongs to and what is their dietary composition also. I can simply apply barcode CY gene to see what kind of animal's fecal is there, and then I can extract the entire plant. I can extract the entire DNA and use the plant primers to see what kind of plants the animal is feeding on or what kind of animal the animal is feeding on. It can be decoded. Okay. Similarly, simply with the fecal material, what see, many bats are actually red listed or you are not supposed to catch them. So in that case, the fecal material can be used to identify the plants, bird species. The fecal material will have a epithelial cells along ejected during the process of excretion, and those cells can and DNA can be recovered from the material, and the CY amplification can be carried out to find the bird species. Yeah, even these days, many interesting articles are done by simply collecting the moisture on the surface of the Amazon forest using drones, and they are decoding the entire forest biodiversity. This is something called non-invasive sampling. Without sampling the plants, the entire diversity can be decoded. Okay, we are also doing such a services. Not only all those gene sequencing services, there is an internship here. So why this internship is every time we hear from our clients that they want to know how to analyze their DNA after they are getting the DNA sequence results from Barcode Biotechnology. They want to know how to analyze the DNA, how to submit it to GenBank, what is the species, how to decode it from the DNA, how to draw a genetic distance data with closely related species, how to make a phylogram. So we thought this can be taught to all the customers or even to the UG students who are interested in DNA sequence analysis. So this workshop, okay. So this is a 14 days intense bioinformatics training where we are looking to analyze DNA sequences. Not only that, they are also providing one free DNA sequencing. Okay, you can send your own sample. That sample will be sequenced, and they are actually using your own machine output file. You can do all the bioinformatics analysis. This is absolutely free. This will give you a real time, real experience of what's happening in the bioinformatics department of Barcode Biotechnology. Okay, not only that, many sequencing companies are very, your results are really delayed only because of this bioinformatics analysis. Many times we see bioinformatics department have very few staff loaded with huge number of sequencing files, and they consume more time. So Barcode Biotechnology is also looking for bioinformatics research associate position. Once you know how to analyze the machine output file and decode the sequences, either in Barcode or many biotech companies involved in DNA sequencing businesses require sequence analytical assistant who can decode the species name from the DNA sequence. That's why this internship. You will hear, I will share the link at below of this video. You can still look into it. Okay, so basically what happens in this internship is the interns will send a courier their sample, animal, plant, microorganism. They create two-way sequencing files, these chromatograms. So in during the internship, they will teach you how to analyze the chromatogram, how to trim out the quality region from the chromatogram, how to trim out your primers from the chromatogram, how to create forward reverse sequences, merge it, how to study each and every peak in the chromatogram. After merging, how to study the mutational rates happen in the genome, or how to compare the gene of a particular species taken from India with that of US or any other geography. In other way of saying, does your DNA have a phylogeographical signal? All those analyses are will be taught how to do it and how to prepare a species identification report. This is a job oriented skill. Okay, when you are given a machine output file, what are the things to be do should be done, and then how to prepare a species identification report that will be mailed to the clients. Okay, and how to merge the sequences, how to subject the sequence to blast analysis, be This is a basic alignment local search tool where your sequence is actually searched with the NCBS database and you can find your species name based on the species cutoff. So how to prepare the sequences of your sequences, say for here, with along with other sequences to check what is the similarity and variation. How to do this multiple sequence alignment at a given position. If there is a star, means all the nucleotides are same. If there is no star, there is a variation in the nucleotide position. So how to do this similarity and variation analysis and how to build a phylogram. When you say your sequence is leuconostoc mesenteroids, it should be placed in the branches of leuconostoc mesenteroids. That's how the phylogram will complement your species identification report. Okay, so all those things are taught, and we will also they are also planning you to teach you about pairwise distance data, how to build this matrix along with phylogram. Okay, and what are the files to be emailed to the clients? All those the industry record skills will be taught. Not only that, they will the program will also teach you how to submit your sequence to NCPI and get an accession number. Okay, so we also very proud about this. Indian Biological Data Center is now available and active. It is an Indian nucleotide data archive where you can submit your sequence and get an Indian accession number, not from US or Japan or in European countries. Now we also have our own database. So barcode gap analysis, this is nothing but the species cutoff I've been talking. In microorganisms, bacteria, we say species cutoff. In macro plants and animals, we'll say barcode gap. Okay. So why this is so important is, see, any manuscript or any publications require a new data to be published. If I'm an editor, I will constantly look for a new data, new findings, so that it get more citation. The scope of running my journal is actually fulfilled. It will be more impactful. They are looking to publish an impactful studies. So whenever I propose a barcode gap, say if my so if whenever I propose a barcode gap, if this is a new gap, which is a new paradigm, my data is a new, I can easily go for publication. Not only that, say many people who went for DNA sequencing do the blast analysis. Whenever they discover their identity is less than the species cutoff, it means that particular species is not there in the database, which qualify them to be submitted as a first barcodes. Okay, if you have this word first barcode, even to impact 2.5 easily can push through. I have seen many paper, even though they don't have a title first barcode, when it is in sequence for first time, it get easily published. Okay. Not only that, even if you have a sequenced species, but if you can define a barcode gap at every taxonomical level, which is an essential parameter now, it is very clear that one parameter is not sufficient. Say one generalized statement of 3% cutoff is not going to work because of various mutational rate and evolutionary constraints. We need to have every hierarchical cutoff. Okay. For example, in this paper I have I have sampled nearly 171 marine fungal samples, which I thought I subjected all 171 for sequencing. Actually, I had nearly 500 specimens, out of which I identified 171 unique morphotypes, which I thought it is 171 species. But when the results come, it was around 54 species belonging to 13 families. So after that, I subjected all the 54 species to blast analysis, collected the top 5 or 10 hits, maximum similarity. I prepared a table. I also discovered that among the 54 fungal species, 12 species were barcoded for first time. This is the first important data for the journal to consider. And then comes the individual. I did the genetic pairwise distance analysis. Then I proposed for marine fungi, 15% can be a cutoff at genera level, 21% cutoff at family level, 30% cutoff at order level, 35 at class level, and 51 at the phyla level. Why this is important is these days meta barcoding, eDNA barcoding is picking up. So if I have I can simply from the air, I can collect the filter through filter paper. I can extract DNA from the filter paper, whatever particles, spores that is being filtered, DNA can be extracted. If I will go for ITS sequencing and do the blast analysis, if my sequence shared only 86% similarity with Aspergillus niger, it means it is a different genera, not even, it can be an Aspergillus genera, sorry, but definitely not niger because the genera cutoff is 15%. If it is 84% similar to Aspergillus niger, definitely it is not Aspergillus and niger. Okay, they may belong to the same Aspergillus A family because it has a 16%. The family cutoff is 21. Okay. So till 79% similarity is allowed. So someone has to define barcode gap at every hierarchy level for better interpretation of new sequences. So this is another new data I find. If you are either you go for first barcoding species when you select your free specimen for this internship, or you just go for pair resistance analysis, have a new data and you can simply publish it. Okay, it is very easy to publish. Here is another work I did. I just took a 20 minutes to see what are the endemic plants which are localized to India. I prepared this 20 list. All red color plant species are not found in the gen bank. I simply put the nucleotide name. I just put their word. I found it is not found. Whenever you see something is not there, it is easy to sequence it and go for a publication. I hope I have done some justice to what is this bacterial species level identification, how it is done actually in the business environment. Okay, so apart from all these sequencing services, they are also involved in skilling the student, because like I said, there is something called Thousand Barcode Initiative where they are constantly looking to enrich the local library with more number of species. Okay, so that EDN and other business aspects can be complemented out of it. So they have a business motive behind this also. So that's why this internship, they have a small fee as shown in the poster, and you are most welcome to send a free sample for sequencing. Before the internship, they can do the sequencing. I guess the internship date has been extended, the registration date has been extended till 22 8 p.m. till tomorrow 8 p.m. So not only this, there is another upcoming internship, because they have a separate division to do all those things. They are actually starting on July. Okay, this is a complete set of internship where they are going to focus on bacterial gene analysis. Okay, both RPOB, that is functional gene, and also the structural genes like 6-ness rRNA gene. And they will also be exploring other barcodes. And like I said, Shigella and E. coli, they are very much genetically similar. 3% cutoff definitely do not work for them. Okay, Shigella, even they have 99.5% similarity with E. coli 6-ness rRNA. So for these peculiar cases, we require to go for a better barcode region. Okay, or better species gap needs to be proposed. How? By aggregating all the genetic data available in the NCBI, and I would suggest take those kind of internships where you'll have a data at the end of it and go for publication. Hope I have done little justice to whatever the topic we have chosen. Uh, Yes, this is another fantastic initiative for Barcode in biotechnology. These are yesterday it has been released. Congratulations to Mahashri, Manjula Devi, and Mother Fatima. They have been qualified for 100% fellowship under this internship, meaning they are going to get a one sequencing service out of free, and they have a 100% scholarship. They are not going to pay anything for this internship, so they will be getting a certificate, and also they will be learning how to analyze the sequences, and they will be given preference when they are appearing for hiring process where there is a remote job of bioinformatics research associate. Okay, so for this seminar, the certificate link will be posted at the end. Right now I'm going to post it, and there will be a quiz. The link will be posted now, and the quiz link will be available. Both the links will be there in the comment section of the video for next 30 minutes. So please use the link to get all your e-certificates. And if you have any questions, there will be a space in the link in the Google form to enter your questions. Please do so, or leave the questions in the comment section. I will comment it, and I will try to answer all your questions. And yes, coming to the quiz part, those who are winning the quiz of from the today's seminar, they will be given one free sequencing of their DNA, whatever that could be, animal, plant, or microorganisms. They can send the samples to us. We will prepare a scholarship certificate for the winners, and we will announce it. You can get the free sequencing out of by securing the topmost position in the quiz. Okay. So that's it for now. Thanks for joining. Have a wonderful day.